Hello there, and welcome to Sleepy Answers. I'm so glad you found your way here tonight. Maybe you've had one of those days where the big questions feel closer than usual, pressing against the edges of your thoughts as evening settles in. Or perhaps you're simply the kind of a person who finds comfort in exploring life's deepest mysteries as you drift toward sleep, who prefers wrestling with the profound, over-counting imaginary sheep. Either way, you're in exactly the right place. Tonight, we're going to contemplate something that touches every living thing that has ever existed, from the smallest bacterium to the largest blue whale, from ancient trilobites to future beings we can't yet imagine. We're going to ask a question that is both utterly ordinary and profoundly strange, both simple and impossibly complex. Why do we die? It's a question that seems straightforward when you first ask it. We die because our bodies wear out, because time takes its toll, because that's just what happens to living things. But the deeper you go, the more layers you discover. There's the biology, the intricate machinery of cells and molecules that eventually winds down like clockwork running out of spring. And there's the chemistry, the reactions and processes that accumulate errors and damage. There's the evolution, the deep logic explaining why nature built us with expiration dates. And then there's the philosophy, the meaning we make of our finite existence, the way mortality shapes how we understand ourselves and our place in the universe. Some people find thinking about death unsettling, even frightening, and that's understandable. We're wired to avoid death, to flinch away from reminders of our mortality. But tonight, we're going to approach it differently. We'll look at death not as something terrible to be feared, but as something fascinating to be understood. We'll examine it from every angle, from the molecular to the metaphysical, and perhaps in understanding it better, we'll find a kind of peace with it. Or at least we'll have some interesting things to think about as we drift off to sleep. Before we begin this exploration, I want to mention that if these kinds of thoughtful, calming journeys through fascinating topics help you unwind at the end of your day, if they give you something substantive to think about as you settle in for the night, please consider liking this video and subscribing to the channel. It helps others who are searching for the same kind of peaceful, curious rest find their way here too. One sleepy soul at a time, we're building a community of people who want to end their days not with mindless distraction, but with meaningful contemplation. So settle in now. Get comfortable. Let your breathing slow and deepen. Your mind can wander through these ideas without grasping too tightly at any of them. We're not here to find definitive answers or to solve ancient mysteries. We're here to explore, to wonder, to turn these questions over in our minds like smooth stones in our hands, feeling their weight and their texture. The question of why we die has occupied human thought for thousands of years. Philosophers have pondered it. Scientists have studied it. Poets have written about it. And everyone who has ever lived has confronted it in some form. Tonight, we'll walk gently through what we've learned, through what we think we understand, and through what remains beautifully mysterious. The universe, as you know, is full of change. Nothing stays the same. Stars are born in cosmic nurseries, burning bright for millions or billions of years before they die in quiet fading or spectacular explosion. Mountains rise slowly from tectonic forces and then erode grain by grain back into the sea. Rivers carve new paths through stone that seems eternal but isn't. Seasons cycle. The moon waxes and wanes. Everything flows. Everything changes. Nothing remains exactly as it was. And living things, from the smallest bacteria to the largest whales, follow the same pattern. They emerge. They exist for a time. They grow and change and interact with their environment, and then they end. But unlike stars or mountains or rivers, living organisms have a special, peculiar relationship with endings. We carry the mechanisms of our own mortality within us, written into the very fabric of our cells, encoded in our genes, built into the fundamental architecture of how life works. It's not something that happens to us from outside. It's something we do to ourselves. Or, rather, something our bodies do as a consequence of being alive in the first place. Let's begin there, at the smallest scale where we can see it happening, where the biological story of death begins in the intricate machinery of the cell. Your body, right at this moment as you lie here listening, is made of roughly 37 trillion cells. That's a number so large it's hard to truly comprehend. If you tried to count them one per second, it would take you over 1,000,000 years. These cells come in hundreds of different types. There are nerve cells with long branching extensions that carry electrical signals across vast distances at the scale of the cell. There are muscle cells packed with proteins that slide past each other to generate force. There are bone cells that secrete mineral deposits, building the scaffold that holds you upright. There are immune cells patrolling your body like microscopic security guards. There are cells lining your intestines, absorbing nutrients from your food. There are cells in your liver, detoxifying chemicals. There are cells in your skin, forming a barrier between you and the world. Each type specialized, each doing its specific job, all working together to create the emergent phenomenon that is you. And right now, as you listen to these words, millions of those cells are dividing, creating copies of themselves to replace the ones that have worn out or been damaged, or simply reached the end of their useful life. In your bone marrow, new blood cells are being born constantly, replacing the 2 million red blood cells that die every second. In your intestinal lining, cells are being replaced every few days because the harsh acidic environment damages them quickly. In your skin, cells are dividing to replace the ones that flake off, the ones you leave behind on everything you touch, even in tissues that seem stable, like your liver or your lungs. Cells are slowly being replaced, maintaining the structure and function of the organs over time. This process of cellular division, this constant renewal, has been happening since you were a single cell, a fertilized egg that divided into 2 cells, then 4, then 8, then 16, doubling again and again in a geometric progression that eventually created the trillions of cells that make up your body. And it continues every moment of your life, a background hum of growth and replacement that you never notice, but that is absolutely essential to your continued existence. But here's the thing, the crucial fact that makes death inevitable. This process can't go on forever. There's a limit built into the system, a cellular countdown clock that ticks with every division. The machinery that allows cells to divide also contains the seeds of its own failure. Understanding this limit, understanding why cells eventually stop dividing and tissues eventually lose their ability to renew themselves, is key to understanding why we age and why we die. Let me take you even deeper, down to the level of individual chromosomes inside your cells. Picture, if you will, the nucleus of a cell, the command center where genetic information is stored. Inside that nucleus are 46 chromosomes, 46 long molecules of DNA, wrapped up with proteins into X-shaped packages. These chromosomes contain all the information needed to build and run your body, written in a code made of just 4 chemical letters: A, T, G, and C. Adenine, thymine, guanine, and cytosine. The particular sequence of these letters, stretching across 3 billion positions in your genome, determines everything from your eye color to your susceptibility to certain diseases to aspects of your personality and intelligence. At the very tip of each chromosome, at the end of each arm of the X, there are structures called telomeres. If you were to zoom in close enough, you'd see that telomeres are not genes that code for proteins. They're not instructions for building anything. They're repetitive sequences, the same 6 letters over and over and over. T-A-G, 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 thousands of times in a row. Think of them as the plastic caps on the ends of shoelaces, the aglets, except instead of preventing the lace from fraying and unraveling, telomeres protect your genetic material from damage and degradation. They're sacrificial buffers, expendable sequences that can be lost without losing any actual genetic information. And they are lost, inevitably, with every cell division. Here's why. When a cell divides, it first has to copy all its DNA so that each daughter cell gets a complete set of genetic information. The molecular machinery that performs this copying, an enzyme called DNA polymerase, works by grabbing onto the DNA strand and moving along it, reading the sequence and building a complementary copy. But there's a problem. A fundamental constraint imposed by the chemistry of how DNA polymerase works. It can't start copying at the very beginning of a strand. It needs a little primer, a short piece of RNA to get started. And it can't copy all the way to the very end. There's always a small segment at the tip of each chromosome that doesn't get copied. This is called the end replication problem. And it was first recognized and theorized by a brilliant Soviet scientist named Alexei Olovnikov back in 1971, decades before we had the experimental tools to directly observe it happening. Olovnikov realized that with each cell division, each round of DNA replication, a little bit of the chromosome end would necessarily be lost, like a candle burning down bit by bit with each lighting, or like a pencil eraser wearing away with each use. And indeed, when scientists in the early 1990s were finally able to measure telomere length in cells, they confirmed that this is exactly what happens. With each division, telomeres get a little shorter, losing about 50 to 200 base pairs each time. After about 50 to 60 divisions, depending on the cell type and various other factors, the telomeres become critically short, worn down to the point where they can no longer protect the chromosome properly. When this happens, the cell recognizes that something is very wrong. Short telomeres look to the cell's DNA damage detection systems, like broken chromosomes. And broken chromosomes are dangerous. They can fuse together, creating genetic chaos. They can lose important genetic information. The cell responds to critically short telomeres by activating a stress response, a molecular alarm system that triggers a permanent shutdown of the cell division process. The cell enters a state called senescence, and it will never divide again. A senescent cell is not dead, not in the traditional sense. It's still metabolically active, still consuming nutrients and producing energy and carrying out many cellular functions. But it has permanently exited the cell cycle. It will never replicate its DNA, never divide into two daughter cells. It's retired, put out to pasture, manim andaner, permanently stuck in a non-dividing state. These senescent cells were first described in the early 1960s by two researchers named Leonard Hayflick and Paul Moorhead, working at the Wistar Institute in Philadelphia. They were studying human cells growing in petri dishes in the laboratory, and they noticed something strange. The cells would divide happily for a while, growing and multiplying as you'd expect. But after somewhere between 40 and 60 divisions, they would stop. The culture would cease to grow. The cells were still alive, still present, but they had lost the ability to divide. This limit became known as the Hayflick limit, after Leonard Hayflick, who characterized it so carefully. For decades, scientists wondered what caused it. Why would cells just stop dividing after a certain number of replications? What was counting the divisions? The discovery of telomere shortening decades later provided the answer. Telomeres are the molecular clock, the counter that keeps track of how many times a cell has divided, and when the count gets too high, when the telomeres get too short, the alarm goes off and division stops. But senescent cells don't just sit there quietly doing nothing. They actually undergo dramatic changes. They swell up, becoming larger and flatter than normal cells, taking up more space in the tissue. Their internal structures reorganize in characteristic ways. Most importantly, they start secreting a cocktail of molecules into their surroundings, a mix of inflammatory signals, growth factors, and enzymes that can break down the structural proteins in tissues. This phenomenon is called the senescence-associated secretory phenotype, often abbreviated as SSP, and it's one of the key ways that aging happens not just at the cellular level, but at the tissue and organ level. Imagine you're in a neighborhood where one or two houses have fallen into disrepair. The owners have moved out but haven't sold the properties. The yards are overgrown, the paint is peeling, windows might be broken. At first, with just one or two such houses, the neighborhood remains functional and pleasant. But as more and more houses fall into disrepair, they start affecting the ones around them. Overgrown yards spread weeds to neighboring properties. Broken windows invite vandalism that might spread. Property values drop, making it harder for people to maintain their homes. Eventually, the entire neighborhood can decline. Not because every single house is in disrepair, but because the accumulation of problem houses creates an environment where decline becomes self-reinforcing. Senescent cells work somewhat like this in your body. One or two senescent cells in a tissue don't cause much of a problem. Your immune system can clear them away, removing them before they cause damage. But as you age, several things happen. First, more cells reach their Hayflick limit and become senescent simply because they've divided many times over your lifetime. Second, other forms of stress damage, oxidation, inflammation can also drive cells into senescence, even if their telomeres aren't critically short. And third, your immune system becomes less efficient at clearing senescent cells away. The result is that senescent cells accumulate in your tissues over time. In young people, senescent cells are rare, quickly dealt with by a vigilant immune system. But in older people, they can make up a significant fraction of cells in some tissues. They accumulate in your skin, contributing to wrinkles and age spots. They accumulate in your joints, contributing to arthritis and loss of mobility. They accumulate in your blood vessels, contributing to atherosclerosis and cardiovascular disease. They accumulate in your fat tissue, your liver, your kidneys, your eyes, your brain. And wherever they accumulate, their inflammatory signals create an environment of chronic low-level inflammation, what scientists sometimes call inflammaging, a combination of inflammation and aging. This chronic inflammation contributes to many of the diseases and conditions we associate with old age, not just arthritis and atherosclerosis, but also diabetes, osteoporosis, neurodegenerative diseases, even cancer. Senescent cells create an environment that makes these diseases more likely to develop and progress. They are, increasingly, thought to be not just markers of aging, but active drivers of it. Not just passengers along for the ride, but pilots steering tissues toward dysfunction. Now, here's where the story gets even more interesting and complex. Telomere shortening is not the only thing that can trigger cellular senescence. Recent research has revealed that telomeres can also be damaged directly, even without shortening. Oxidative stress, the kind of molecular damage caused by reactive oxygen species produced during normal cellular metabolism, can damage telomeres. DNA breaks or chemical modifications to the telomeric DNA can make telomeres dysfunctional even if they're still technically long enough. This means that telomeres act not just as a division counter, but as a sensor of overall cellular health and stress. A cell that has been exposed to a lot of oxidative damage or to other forms of stress may become senescent even if it hasn't divided very many times. The telomeres, in effect, are saying, "This cell has experienced too much stress. It's at risk of becoming dysfunctional or dangerous. Time to shut it down." This broader sensing function makes telomeres even more central to the aging process than scientists initially realized. And there's another key player in this story that we need to talk about, an enzyme called telomerase. This remarkable molecular machine has the ability to add DNA sequences back onto telomeres, essentially resetting the molecular clock, or at least slowing it down. Telomerase was discovered in the late 1980s by Carol Greider and Elizabeth Blackburn, working with a single-celled organism called tetrahymena. They showed that this enzyme could extend telomeres by adding new TTAGG repeats, counteracting the shortening that occurs with each cell division. This was a huge discovery, important enough that Grider and Blackburn received the Nobel Prize in Physiology or Medicine in 2009. It revealed that telomere shortening isn't an absolutely immutable fact of cellular life. There is a mechanism to counteract it. The question then becomes, if we have this mechanism, why do our cells still experience telomere shortening? Why doesn't telomerase just keep our telomeres long indefinitely? The answer is that in most adult human cells, telomerase is turned off. The gene that codes for the catalytic component of telomerase, called TERT, is not expressed in most of your body's cells. There are important exceptions. Stem cells, which need to divide many times over your lifetime to replenish various tissues, have active telomerase. Reproductive cells, the ones that will eventually form eggs or sperm, have active telomerase because they need to pass undamaged, full-length telomeres on to the next generation. But regular somatic cells, the ordinary cells that make up your skin and muscles and organs, don't have telomerase activity. Their telomeres shorten with each division, and eventually they hit the Hayflick limit. Why would your body be designed this way? Why would we have a mechanism to extend telomeres, but deliberately keep it turned off in most cells? The answer lies in the dangerous relationship between unlimited cell division and cancer. Cancer is fundamentally a disease of cells that have lost the normal controls on cell division. A cancer cell divides when it shouldn't, ignores signals telling it to stop, and eventually forms a tumor. For a cancer to grow large and dangerous, it needs to overcome the Hayflick limit. If cancer cells divided only 40 or 50 times, tumors would remain small and much less dangerous. And indeed, one of the hallmarks of cancer cells is that they've reactivated telomerase. In about 90% of human cancers, the cancer cells have high levels of telomerase activity, allowing them to divide indefinitely, to keep growing and spreading without hitting the cellular limit that would normally stop them. The other 10% of cancers use alternative mechanisms to maintain their telomeres, but the principle is the same. To become truly dangerous, cancer cells need to become immortal at the cellular level. Telomere shortening, then, acts as a powerful tumor suppressor mechanism. It's a safeguard, ensuring that if a cell starts dividing uncontrollably due to mutations or other damage, it will eventually hit a limit and stop. The cell will become senescent before it can form a large tumor. This protection isn't perfect, obviously. Cancer still occurs, but it's significant. Without this mechanism, cancer would be far more common and far more dangerous than it already is. The price we pay for this protection is aging. Our bodies have essentially made a trade-off, choosing a strategy that prevents most cancers during our reproductive years and through midlife, but guarantees that our cells will eventually lose their ability to divide, and our tissues will eventually lose their ability to regenerate properly. It's a compromise, a balancing act between the risk of cancer and the process of aging. And it's written into our biology at the most fundamental level, into the structure of our chromosomes and the regulation of our genes. This trade-off between cancer protection and aging is an example of a broader principle in the biology of aging called antagonistic pleiotropy. The term sounds complicated, but the concept is straightforward. pleiotropy means that a single gene or mechanism can have multiple effects. Antagonistic pleiotropy means that those effects can be in opposition to each other, with some being beneficial and others being harmful. Specifically, in the context of aging, antagonistic pleiotropy refers to genes or mechanisms that are beneficial early in life but harmful later on. Evolution, remember, doesn't care about your comfort in old age. It cares about reproductive success. Natural selection strongly favors traits that help you survive to reproductive age, find a mate, successfully reproduce, and raise offspring to the point where they can reproduce. After that, from evolution's cold perspective, you're less important. You've already passed on your genes. Whether you live in good health for decades beyond that or not matters much less to evolution. Genes, or mechanisms that provide advantages early in life, will be selected for even if they cause problems late in life, because those problems occur after the individual has already reproduced. If a gene makes you more resistant to infection as a child, increasing your chances of surviving to adulthood, but also contributes to chronic inflammation in old age, natural selection will favor that gene. The benefit comes at the time when it matters most for reproductive success. The cost comes later, after the genetic dice have already been passed to the next generation. Telomere shortening and cellular senescence fit this pattern perfectly. They protect you from cancer during your youth and reproductive years, when cancer would be most likely to interfere with reproduction. The fact that they lead to tissue dysfunction and aging decades later is, from an evolutionary standpoint, acceptable collateral damage. Your genes have already made it into the next generation. What happens to your body after that is not evolution's concern. This logic extends to many other aspects of aging. Consider the immune system. A vigorous immune response in youth helps you fight off infections that might otherwise kill you before you can reproduce. But that same vigorous immune response, if it persists at high levels into old age, contributes to chronic inflammation and autoimmune problems. Or consider the stress response. The ability to mount a strong stress response, releasing hormones like cortisol that mobilize energy and sharpen your senses, is valuable when you're facing immediate threats. But chronic elevation of stress hormones in old age contributes to numerous health problems. These are all examples of mechanisms that are useful, even necessary, in youth, but that become problematic if they persist or accumulate in old age. And because natural selection is much stronger in youth than in old age, evolution tends to optimize for youth at the expense of old age. We are, in a very real sense, built to reproduce, not to live forever. This brings us to another fundamental theory of aging, one that explains not just why individual mechanisms might fail, but why evolution hasn't simply built us to last longer. This theory is called the disposable soma theory, proposed by Thomas Kirkwood in the 1970s. Soma, in this context, refers to your body, your somatic cells, as distinguished from your germline cells, the reproductive cells that will contribute to the next generation. The disposable soma theory starts with a simple observation. Organisms have limited resources, limited energy, limited nutrients. These resources must be allocated between different biological priorities. Two of the major priorities are maintenance and reproduction. Maintenance means keeping your body in good working order, repairing damage, replacing worn-out parts, defending against disease. Reproduction means producing offspring and, in many species, caring for them until they can survive independently. Here's the key insight. There's a trade-off between these priorities. Resources spent on maintenance are not available for reproduction. Resources spent on reproduction are not available for maintenance. An organism that invests heavily in keeping its body in perfect condition for many decades, but reproduces slowly, will be out-competed by organisms that reproduce more quickly, even if those organisms wear out faster. In the competitive arena of evolution, the strategy that produces more offspring in the available time tends to win, even if it means sacrificing longevity. Think of it like running a business with a limited budget. You can invest in maintenance and quality control, making sure all your equipment lasts a long time and your products are built to be durable. Or you can invest in production and marketing, churning out more products and getting them to more customers, even if the products aren't built to last as long. In a competitive market, the business that prioritizes production and market share often wins out over the business that prioritizes durability and longevity, because it can establish dominance before the slower competitor has a chance to scale up. Evolution works similarly. Organisms that put more resources into reproduction, even at the cost of long-term body maintenance, tend to have more offspring, and thus spread their genes more successfully. Over evolutionary time, this leads to life strategies where the soma, the body, is treated as somewhat disposable. It needs to last long enough to reproduce successfully, but it doesn't need to last forever. In fact, investing too much in keeping it in pristine condition would be a waste of resources that could have gone into reproduction. Different species, of course, make different trade-offs based on their ecological circumstances. Small mammals, like mice, face high predation risk. They might get eaten at any moment. Under these conditions, evolution favors a fast life strategy. Grow quickly, reproduce early and often. Don't bother investing too much in long-term maintenance because you probably won't live long enough for it to matter. Mice typically live only a year or two, even in protected laboratory conditions where there's no predation. They're built for rapid reproduction, not longevity. Larger mammals, which face less predation, can afford to invest more in maintenance and longevity. Elephants, for example, live 50 to 70 years. They grow slowly, reproduce infrequently, and invest heavily in caring for their young. This is a slow life strategy, made possible by their size and relatively low predation risk. They can afford to build bodies that last because they're likely to live long enough for that investment to pay off reproductively. Birds and bats, which can fly and thus escape many predators, tend to live longer than similar-sized mammals that can't fly. The ability to escape predators makes investing in longevity more worthwhile. Naked mole rats, peculiar rodents that live in underground colonies with low predation risk and stable environmental conditions, can live over 30 years, which is extraordinarily long for a rodent their size. Their protected lifestyle makes a slow life strategy viable. Humans fall somewhere in the middle, but leaning toward the slow end of the spectrum. We're large, relatively long-lived mammals with big brains, extended childhoods, and complex social structures. Our evolutionary history as social primates with low predation risk and high parental investment has shaped us for a life strategy that includes significant investment in body maintenance. We can live 70, 80, 90 years or more, but we're still mortal. We still age. Evolution still treated our bodies as temporary vehicles for our genes, not as ends in themselves. All of this, the telomeres and cellular senescence, the evolutionary trade-offs, the limited resources allocated to maintenance versus reproduction, explains why we age at the cellular and tissue level. But there are other mechanisms of aging happening simultaneously, other molecular clocks ticking down, other forms of damage accumulating. Understanding these other mechanisms gives us a more complete picture of why aging is so complex and why solving it is so difficult. Consider mitochondria, those tiny power plants inside your cells that generate most of your body's energy. Every cell in your body except red blood cells contains hundreds or even thousands of mitochondria. These organelles are fascinating in their own right because they're not truly part of your cells. They're the descendants of ancient bacteria that were engulfed by ancestral cells billions of years ago and evolved into a symbiotic relationship. They still have their own DNA, separate from the DNA in your cell nucleus, and they still reproduce independently, dividing and fusing within your cells. Mitochondria generate energy through a process called oxidative phosphorylation, which involves moving electrons along a chain of protein complexes embedded in the mitochondrial membrane. As electrons move along this chain, protons are pumped across the membrane, creating a concentration gradient. The energy stored in this gradient is then used to synthesize ATP, the molecule that powers most cellular processes. It's an elegant system, refined over billions of years of evolution, and it's remarkably efficient. But it's not perfectly efficient. Occasionally, electrons escape from the electron transport chain before they're supposed to. These free electrons react with oxygen molecules, creating reactive oxygen species, highly reactive molecules that include superoxide radicals, hydrogen peroxide, and hydroxyl radicals. These are what people commonly call free radicals, though that term is somewhat imprecise. Reactive oxygen species are dangerous because they can react with and damage almost anything they encounter. They can damage proteins, causing them to lose their shape and function. They can damage lipids in cell membranes, creating holes and making membranes leaky. And, crucially, they can damage DNA, causing mutations and breaks in the genetic code. This includes the mitochondrial DNA itself, which is particularly vulnerable because it's right there in the mitochondria where the reactive oxygen species are being produced. And because mitochondrial DNA doesn't have the same protective packaging and repair systems that nuclear DNA does. As mitochondrial DNA accumulates mutations and damage over time, the mitochondria become less efficient at producing energy, and less efficient mitochondria produce even more reactive oxygen species for every unit of energy they generate. It's a vicious cycle. Damaged mitochondria produce more oxidative stress, which causes more damage to mitochondria, which produces more oxidative stress. Over time, this progressive mitochondrial dysfunction contributes to cellular aging and the decline of energy-intensive tissues like brain, heart, and skeletal muscle. For decades, this free radical theory of aging, first proposed by Denham Harmon in the 1950s, was one of the dominant explanations for why we age. The idea was appealingly simple. Aging is caused by the accumulation of oxidative damage from free radicals over time. If we could reduce free radical production or increase the body's ability to neutralize them, we might slow aging. But as with many simple, appealing ideas, the reality turned out to be more complicated. Studies that gave animals antioxidants, compounds that can neutralize free radicals, didn't always extend lifespan, and sometimes even shortened it. Mice, genetically engineered to produce extra antioxidant enzymes, didn't always live longer. It became clear that reactive oxygen species aren't purely harmful. They also play important roles as signaling molecules, helping to regulate various cellular processes including stress responses and even defense against infections. The current understanding is more nuanced. Oxidative stress certainly contributes to aging, but it's not the single primary cause. It's one factor among many, interacting with other mechanisms in complex ways. Chronic, excessive oxidative stress is harmful and does contribute to cellular dysfunction and disease. But some level of reactive oxygen species production is normal and even necessary. The relationship between oxidation and aging is not as simple as more oxidation equals more aging. Importantly, oxidative stress interacts with telomeres and cellular senescence. We now know that oxidative damage to telomeres can trigger senescence even in cells that haven't divided many times. This means that mitochondrial dysfunction, by increasing oxidative stress, can accelerate telomere dysfunction and cellular senescence, linking these two major mechanisms of aging together. Another clock ticking inside your cells involves proteins themselves. Your body contains tens of thousands of different types of proteins, each one a precisely folded chain of amino acids that performs a specific function. Some proteins act as enzymes, catalyzing chemical reactions. Others form structural scaffolds, giving cells their shape. Others transport molecules, or receive signals, or regulate genes. Proteins are the workhorses of the cell, doing essentially all the actual work of keeping you alive. But proteins are delicate. They need to be folded into exactly the right three-dimensional shape to function properly, and that shape can be disrupted by all sorts of things: heat, oxidation, random chemical reactions, interactions with other molecules. When a protein misfolds, it usually stops working. Even worse, misfolded proteins can clump together, forming aggregates that are not just non-functional but actively toxic to cells. Your cells have elaborate systems to deal with protein quality control. Chaperone proteins, aptly named, help other proteins fold correctly when they're first made, and can help refold proteins that have become partially unfolded. If a protein is damaged beyond repair, it gets tagged for destruction and broken down by cellular machines called proteasomes, or engulfed by structures called lysosomes. The components are then recycled to make new proteins. These quality control systems work well when you're young. Damaged proteins are quickly identified and removed. Protein aggregates are cleared before they accumulate. But with age, these systems become less efficient. Chaperones don't work as well. proteasomes slow down. Lysosomes become clogged with junk they can't fully digest. The result is that misfolded and aggregated proteins begin to accumulate in your cells and tissues. In some tissues, particularly the brain, this accumulation of protein aggregates is associated with serious age-related diseases. Alzheimer's disease involves accumulation of two types of protein aggregates: plaques made of beta-amyloid protein outside neurons, and tangles made of tau protein inside neurons. Parkinson's disease involves accumulation of aggregates of a protein called alpha-synuclein. Other neurodegenerative diseases have their own characteristic protein aggregates. These aggregates are not just markers of disease but appear to be actively toxic, interfering with cellular function and contributing to the death of neurons. But protein aggregation isn't just a problem in dramatic diseases. Even in normal aging, in people who don't develop Alzheimer's or Parkinson's, there's a gradual accumulation of damaged and aggregated proteins throughout the body. This contributes to the general decline in cellular function that characterizes aging. Cells full of protein junk simply don't work as efficiently as clean, young cells. Then there's the DNA itself, your genetic material, your instruction manual for life. You might think that DNA, being so important, would be perfectly stable and protected, but in fact, your DNA is under constant assault. Every single day, every cell in your body experiences thousands of damaging events to its DNA. Cosmic radiation streaming down from space creates breaks and mutations. Ultraviolet light from the sun damages DNA in your skin cells. Reactive oxygen species, those same free radicals we talked about, damage DNA throughout your body. Even the process of copying DNA during cell division, despite being quite accurate, makes occasional errors. Fortunately, your cells have elaborate DNA repair systems, multiple pathways with different enzymes dedicated to fixing different types of damage. When a base in the DNA is chemically modified, base excision repair removes it and replaces it with the correct base. When there's a break in one strand of the DNA double helix, Nick-sealing enzymes close the gap. When both strands are broken, more complex repair mechanisms glue the ends back together. These repair systems are remarkably good at their job. Most DNA damage is fixed correctly with no lasting consequences. But the repair systems aren't perfect. They occasionally make mistakes, fixing damage incorrectly or incompletely. And importantly, like other cellular systems, DNA repair becomes less efficient with age. Repair enzymes are expressed at lower levels. The pathways don't work as quickly. The result is that unrepaired or incorrectly repaired DNA damage accumulates over your lifetime. This accumulation of DNA damage has several consequences. Some damaged cells die through a process called apoptosis, programmed cell death. This is actually a good outcome, removing potentially dangerous cells before they can cause problems. Other cells with DNA damage become senescent, entering that permanent non-dividing state we talked about earlier. Again, this prevents the cell from continuing to divide with damaged DNA, but contributes to tissue aging through the accumulation of senescent cells. And some cells with DNA damage continue to divide, carrying mutations into their daughter cells. Most of these mutations are neutral or mildly harmful to the cell, slightly reducing its function but not causing any major problems. But occasionally, a mutation hits a critical gene, one involved in controlling cell division or responding to growth signals. If the right combination of mutations accumulates in the same cell, that cell can become cancerous, dividing uncontrollably and potentially forming a tumor. This is why cancer risk increases so dramatically with age. It's not just that we've been exposed to more carcinogens over time, though that's part of it. It's also that our cells have accumulated more mutations over time, making it more likely that the particular combination of mutations needed to create a cancer cell will occur. And it's that our DNA repair systems, our immune surveillance, and other protective mechanisms have all become less efficient, making it easier for a cancer cell to escape detection and grow into a tumor. Beyond the sequence of DNA bases themselves, there's another layer of information encoded in your genome, the epigenome. This refers to chemical modifications on the DNA and on the histone proteins the DNA wraps around, modifications that control which genes are turned on and which are turned off. Epigenetic marks are crucial for cellular identity. A skin cell and a liver cell have the same DNA sequence, but different patterns of epigenetic marks, which is why they look different and do different things. These epigenetic patterns are maintained as cells divide, passed on from mother cell to daughter cells, preserving cellular identity across generations. But with age, epigenetic patterns begin to drift. Marks that should be in certain places are lost. Marks appear in places they shouldn't be. The result is a gradual loss of proper gene regulation. Genes that should be on get turned off. Genes that should be off get turned on. This epigenetic noise, this growing disorder in the regulatory system, contributes to the loss of cellular function with age, to cells not being quite as good at being what they're supposed to be. So we have multiple mechanisms of cellular aging, multiple clocks running at different speeds, telomere shortening leading to cellular senescence, mitochondrial dysfunction increasing oxidative stress, protein aggregation clogging cellular machinery, DNA damage accumulating mutations, and epigenetic drift disrupting gene regulation. And, crucially, these mechanisms aren't independent. They interact in complex ways, forming a web of causes and effects that amplify each other. Mitochondrial dysfunction increases oxidative stress. Oxidative stress damages telomeres, proteins, and DNA. DNA damage can trigger senescence. Senescent cells secrete inflammatory factors. Inflammation increases oxidative stress. The whole system is interconnected, with problems in one area creating problems in others, a cascade of dysfunction that accelerates over time. And all of this is happening while your body's capacity for renewal is declining. Many of your tissues contain populations of stem cells, special cells that remain relatively undifferentiated and can divide to produce new specialized cells when needed. Stem cells are what allow your bone marrow to keep producing new blood cells, your intestinal lining to keep regenerating, your liver to repair damage, your muscles to grow and adapt. But stem cells are also subject to all the same mechanisms of aging we've been discussing. their telomeres shorten. Their DNA accumulates damage. They become senescent. As stem cell populations decline and their function deteriorates, tissues lose their ability to repair and regenerate properly. Your skin becomes thinner and less elastic because the stem cells that produce new skin cells are running low and not working as well. Your muscles lose mass and strength, a condition called sarcopenia, in part because muscle stem cells become less able to repair and rebuild muscle tissue. Your bones become more fragile because the stem cells that produce new bone cells are failing to keep pace with bone loss. This decline in regenerative capacity is one of the most visible and consequential aspects of aging. It's what turns minor injuries that would have healed quickly in youth into serious problems in old age. It's what makes recovery from illness or surgery so much slower. It's what creates the overall fragility that characterizes advanced age. Now, at this point, you might be wondering, if all these mechanisms of aging are problems, why hasn't evolution simply fixed them? Why doesn't natural selection weed out genes that cause aging and favor genes that prevent it? If living longer would allow an organism to have more offspring over its lifetime, shouldn't evolution favor longevity? The answer brings us back to the fundamental evolutionary logic we touched on earlier, but now we can appreciate it in more depth. Natural selection does not optimize for longevity per se. It optimizes for reproductive success, which is not quite the same thing. In environments where extrinsic mortality is high, where most organisms die young from predation, disease, accident, or starvation, there's little selective advantage to building a body that could last 100 years. That body will almost certainly be killed long before it reaches its potential lifespan. Under these conditions, it makes more evolutionary sense to invest in reproducing as quickly as possible, getting your genes into the next generation before something kills you, rather than investing heavily in maintaining your body for a long lifespan you're unlikely to achieve anyway. This is why in the wild very few animals die of old age. They die of other things first. The intrinsic aging of their bodies rarely becomes relevant. Humans are somewhat special in this regard because we evolved complex social structures and technologies that reduced extrinsic mortality. We learned to use tools, to build shelters, to hunt cooperatively, to care for sick and injured members of our groups. This made it more likely that we would actually live long enough for aging to matter. And indeed, there's evidence that older humans, past their own reproductive years, contributed to the survival and success of their children and grandchildren. This is sometimes called the grandmother hypothesis. The idea that there was evolutionary advantage to living long enough to help raise grandchildren. But even with this, the selective pressure for extreme longevity is weak. Once you've helped your grandchildren to a point where they're likely to survive and reproduce, evolution has little more use for you. The mechanisms that keep you alive and healthy into your 60s and 70s have some selective advantage. The mechanisms that would keep you healthy and vigorous into your hundreds have almost no selective advantage, because very few people throughout human evolutionary history lived that long, and those who did had already passed on their genes and helped raise their descendants. This is why different species age at such dramatically different rates. It's not that the biological problems of aging are fundamentally different across species. Telomeres shorten, proteins misfold, DNA accumulates damage in mice just as in elephants. But the balance of trade-offs, the allocation of resources between maintenance and reproduction, varies based on ecological circumstances. Small mammals with high extrinsic mortality invest little in maintenance and age quickly. Large mammals with low extrinsic mortality can invest more in maintenance and age more slowly. Flying animals, protected from many predators by their ability to escape, tend to live longer than similar-sized animals that can't fly. These differences demonstrate that there's nothing fundamentally inevitable about a particular rate of aging. The machinery exists throughout biology to maintain bodies in good condition for longer periods. Natural selection can tune the rate of aging up or down based on ecological circumstances. But in every case, aging eventually occurs because it's not worth the evolutionary cost to prevent it entirely. This brings us to an interesting question, one that occupies a growing field of biomedical research. If aging is the result of specific biological mechanisms that differ across species and that evolution can modulate, could those mechanisms be modified in humans? Could we slow aging or even stop it entirely? The answer is complex. On one hand, research on taught aging has made remarkable progress in recent decades. Scientists have identified numerous interventions that extend lifespan in laboratory organisms. In yeast, worms, flies, and mice, we can manipulate genes, alter diets, or give drugs that increase lifespan, sometimes dramatically. This proves that aging is not some mystical, untouchable process, but a biological phenomenon that can be modified. One of the most exciting areas of current research involves those senescent cells we discussed earlier. Remember, senescent cells accumulate with age and actively contribute to tissue dysfunction through the inflammatory factors they secrete. Scientists have developed a class of drugs called senolytics, designed to selectively kill senescent cells while leaving normal cells alone. When researchers give senolytic drugs to old mice, remarkable things happen. The mice become more physically active. Their organ function improves. They experience delayed onset of age-related diseases, and in some studies they live longer. Most strikingly, the mice appear in many ways younger, as if removing the senescent cells has partially turned back the clock. These animal studies have generated enormous excitement because they suggest that senescent cells are not just markers of aging, but active drivers of it. Remove them, and tissues function better. Multiple senolytic drugs are now in human clinical trials for various age-related diseases, from arthritis to lung fibrosis to kidney disease. Early results have been promising, with some patients showing improvements in function and reductions in inflammatory markers. But it's important to temper our excitement with realism. Senolytic drugs are still experimental. We don't yet know if they'll be safe and effective for long-term use in humans. We don't know if they'll actually extend human lifespan or just health span, the period of life spent in good health. And we don't know what unforeseen side effects might emerge. Still, this is one of the most promising approaches to targeting aging directly, rather than just treating individual age-related diseases. Another approach involves telomerase, that enzyme that can extend telomeres. If short telomeres drive cellular senescence, and senescence drives aging, could we slow aging by preventing telomere shortening? In mice, experiments have shown that activating telomerase in old animals can reverse some aspects of aging, restoring function to tissues and organs, and even extending lifespan. This is remarkable evidence that telomere shortening is not just a consequence of aging, but a contributor to it. However, there's a major concern. Remember that most cancer cells have reactivated telomerase, and that this is necessary for them to become dangerous. Simply turning on telomerase in all your cells might increase cancer risk unacceptably. Some compounds derived from traditional medicines, like TA65 extracted from a Chinese herb called astragalus, have been shown to have mild telomerase activating effects and are marketed as anti-aging supplements. But their effectiveness in humans is unclear, and their long-term safety is uncertain. The challenge is to find ways to maintain telomeres, or clear senescent cells without increasing cancer risk. This is a delicate balance, given that telomere shortening and cellular senescence are themselves tumor suppressor mechanisms. It's possible that periodic, temporary activation of telomerase, or selective elimination of senescent cells, might provide benefits without unacceptable risks. But we're still figuring this out. Researchers have also discovered metabolic interventions that can slow aging. The most robust and reproducible is caloric restriction, eating significantly fewer calories while still getting adequate nutrition. In organisms from yeast to worms to flies to mice, caloric restriction extends both average and maximum lifespan, often by 20 to 40%. Animals on caloric restriction are healthier, more active, and live longer. The mechanisms are not fully understood but seem to involve activation of stress response pathways that help cells cope with damage, reductions in metabolic rate that decrease production of damaging reactive oxygen species, and shifts in how cells allocate resources. Essentially, when food is scarce, organisms shift from growth and reproduction mode into maintenance and survival mode, investing more in keeping the body in good condition. Does this work in humans? We don't have definitive evidence yet because it would require decades-long studies to find out. But shorter-term studies have shown that caloric restriction in humans does produce many of the same metabolic changes seen in rodents: reduced inflammation, improved insulin sensitivity, lower blood pressure, better cholesterol profiles. Whether it extends human lifespan remains to be proven, but it seems plausible. However, sustained caloric restriction is difficult for most people to maintain. Feeling hungry all the time is unpleasant, and significant caloric restriction can have side effects, including cold sensitivity, reduced libido, loss of bone and muscle mass, and psychological effects. So, researchers have looked for drugs that might mimic the effects of caloric restriction without requiring people to actually restrict their calories. Compounds like rapamycin and metformin, both existing drugs used for other purposes, show some promise. Rapamycin, an immunosuppressant drug, extends lifespan in mice even when started late in life. Metformin, a diabetes drug, has been associated with reduced mortality in some human studies, though whether it truly extends lifespan or just reduces diabetes-related deaths is unclear. Human trials of these drugs for aging are ongoing, but again, it will take many years to get definitive answers. So, where does this leave us? There's real scientific progress happening. We understand the mechanisms of aging far better than we did even 20 years ago. We have interventions that work in laboratory animals. We're beginning to test some of these in humans. It seems likely that in the coming decades, we'll develop therapies that can slow some aspects of aging and extend health span, allowing people to remain healthy and functional longer. But we need to be realistic about the prospects for radical life extension, for dramatically pushing back maximum lifespan or achieving anything like biological immortality. The mechanisms of aging are deeply intertwined and highly complex. Addressing one mechanism often runs into problems with others. Fix telomeres, and you might increase cancer risk. Clear senescent cells, and you might impair wound healing, since senescence plays important roles beyond aging. Boost one pathway, and you might have unforeseen effects on others. More fundamentally, our bodies are the product of millions of years of evolution that optimized us for survival and reproduction in a particular ecological niche, not for indefinite longevity. We are, in a very real sense, built to age. Our design incorporates trade-offs and compromises that made sense in our evolutionary past, but that create inevitable problems over extended time. Redesigning a human being for extreme longevity would require changing fundamental aspects of our biology. Aspects that are interlinked with everything else about how we function. None of this means we can't make progress. It seems quite likely that we'll be able to compress morbidity, reducing the period at the end of life spent in poor health, allowing people to remain vigorous into their 70s, 80s, perhaps even 90s, and then decline relatively quickly. This would be a huge triumph, even if maximum lifespan doesn't change much. The goal isn't necessarily to live to 150, but to live to 80 or 90, feeling like you're 60. But let us step back now from the molecular and cellular level, from talk of telomeres and mitochondria and senolytic drugs. Because death is not just a biological phenomenon, not just something that happens when cells stop dividing and organs stop functioning. Death is also, perhaps primarily, a philosophical problem. It's something we're aware of, something we think about, something that shapes how we understand our existence. And the way we think about death, the meaning we make of our mortality, profoundly affects how we live our lives. One of the earliest and most influential philosophical treatments of death comes from the ancient Greek philosopher Epicurus, who lived in the 3rd and 4th centuries BC. In a letter to a follower named Menoetius, Epicurus argued that death should not be feared because it is nothing to us. This is not a throwaway comment, but a carefully reasoned position. Epicurus's argument goes like this: all good and all evil, everything that can be good or bad for us, consists in sensation, in our ability to experience things. Pleasure is good, pain is bad, and these are defined by the sensations they produce. Death is the complete absence of sensation. When we are dead, we don't sense anything. We don't experience anything. Therefore, death cannot be good or evil for us. It's simply nothing, a blank, a total absence. More specifically, Epicurus pointed out that we cannot actually experience death. While we are alive, death is not present. We're here, experiencing life. And when death is present, when we have actually died, we no longer exist. We're not there to experience anything at all, including the state of being dead. The two states, life and death, never overlap. There is no moment at which we will experience death or the badness of being dead. To make this more vivid, Epicurus compared the time after our death to the time before our birth. We did not exist for billions of years before we were born. The universe carried on without us. Stars formed and died. Galaxies swirled. The earth cooled and developed life. All of this happening during the vast stretch of time before we existed. And that non-existence caused us no distress. We don't look back on all those eons before our birth with any sense of deprivation or loss. They were simply a time when we weren't. Why, then, asked Epicurus, should the non-existence that follows our death be any different? We will not exist for billions or trillions of years after we die. The universe will carry on without us, just as it did before we were born. But that future non-existence should be no more concerning than our past non-existence. Both are simply times when we are not. And neither can harm us because we're not there to be harmed. This argument has a certain elegant logical clarity. If death really is just non-existence, and if non-existence has no experiential content, then rationally, there is nothing to fear. The fear of death, on this view, is a kind of confusion, an irrational response that comes from imagining ourselves somehow experiencing death, when in fact death is precisely the absence of all experience. Death is nothing to us. Therefore, as Epicurus concluded, when properly understood, death should not be feared. This was not just an abstract intellectual position for Epicurus. His philosophy, known as Epicureanism, was explicitly therapeutic, aimed at helping people live happier, more tranquil lives. He thought that much human misery came from irrational fears and desires, and that by thinking clearly about things like death, we could free ourselves from unnecessary anxiety and live with greater peace of mind. The fear of death, for Epicurus, was one of the great sources of human misery, and he offered his argument as a kind of cure. For many people throughout history, Epicurus's argument has been comforting. If you truly accept that death is nothing, that it's not an experience you'll have, but simply an absence, then much of the sting is removed. You don't have to spend your life dreading something that, by definition, you will never experience. But many philosophers, both ancient and modern, have found Epicurus's argument unsatisfying. Even if we grant that the state of being dead is not itself bad, that we won't experience it, and thus can't be harmed by it, doesn't death still seem like a bad thing? Doesn't the fact that we will cease to exist still seem like something worth regretting or fearing? The philosopher Thomas Nagel, in the 20th century, articulated what's called the deprivation account of why death is bad. According to this view, death is bad not because of any positive features of being dead, not because death is something terrible we will experience, but because death deprives us of something valuable, continued life, and all the goods that life would have contained. Think about it this way. If you were to die tomorrow, you would miss out on all the future experiences you might otherwise have had, all the conversations with friends and family, all the beautiful sunsets and delicious meals and interesting books, all the laughter and love and small pleasures that make life worth living, all the achievements and satisfactions, the moments of meaning and connection. These goods would be lost to you, foreclosed, no longer possible. Death, on this account, is bad because it's the permanent end of all future goods. It's a deprivation, a loss. Not a loss you'll experience, granted, since you won't exist to experience the loss, but a loss nonetheless. You will have been deprived of years or decades of life you might otherwise have had, of experiences and relationships and achievements that might have been but now will never be. This seems, to many people, to capture something important that Epicurus's argument misses. Yes, being dead isn't itself a bad experience because it's not an experience at all. But dying is still bad because it ends your opportunity to have experiences. The badness of death lies not in death itself, but in what death takes away from you. However, this leads to some philosophically puzzling questions. If death is bad because it deprives you of future goods, when exactly is it bad for you? Not after you're dead, because you don't exist then. You're not around to be harmed by anything. But not before you die either, because while you're alive, you haven't yet been deprived of future life. You still have it, at least until the moment of death. So the harm of death seems to occur at no particular time, which is philosophically strange. Harms usually happen at some specific time to some specific person. But the harm of death doesn't fit this pattern. Some philosophers have tried to resolve this by arguing that something can be bad for you without occurring at any particular time and without you experiencing it. Being slandered behind your back might be bad for you even if you never find out about it, even if it never directly affects your experiences. Similarly, being deprived of future life might be bad for you, even though you don't exist at the time when you could be said to have been deprived. Others have suggested focusing not on the state of being dead, but on the event of dying, the transition from life to non-life. This event occurs while you still exist, so the harm can be located in time and in a person. Of course, the process of dying often involves suffering, pain, fear, grief, and those things are clearly bad. But even a hypothetical, painless, instantaneous death would still seem bad because it ends your existence. These debates about whether and how death is bad might seem overly abstract, like philosophical games removed from real life. But they matter because how we think about death affects how we feel about it and thus how we live. If we believe Epicurus that death is nothing to fear because it's literally nothing to us, we might live with less anxiety about mortality. If we believe the deprivation account, we might feel our mortality more keenly but also feel more motivated to make the most of the time we have, since every day not lived is a day of experiences lost. The 20th century existentialist philosopher Martin Heidegger offered yet another perspective, one that makes death central to the very structure of human existence. For Heidegger, death is not primarily something that happens at the end of biological life. It's not primarily an event in the future. Instead, death is a constant feature of human existence, something that shapes every moment of our lives, whether we're consciously thinking about it or not. Heidegger used the term being toward death to describe the human relationship with mortality. We are always, in a fundamental sense, moving toward our death. We are beings whose existence will end, and this finitude, this limitation, this guaranteed terminus, pervades everything we are and do. Death is not just something that will happen someday. It's a defining feature of what it means to be human. We are the beings who know we will die. More specifically, Heidegger called death our own most possibility. It's the one possibility that is most uniquely our own, most intimately ours. Everything else about us might be shared with others or transferred to others. Our possessions can be given away. Our roles and responsibilities can be taken up by someone else. But our death is absolutely our own. No one can die for us or in our place. Each of us must, in the end, face our own death alone. Moreover, death is what Heidegger called an absolute possibility. By this he meant that death is not something conditional, not something that might happen. It's certain. We will die. This is not a possibility in the sense of something that might or might not occur. It's a possibility in the sense of something that defines the horizon of our existence, the limit beyond which we cannot go. For Heidegger, how we relate to our own mortality is crucial to whether we live authentically or inauthentically. Most people, most of the time, exist in what he called an inauthentic mode. We lose ourselves in the routines and conventions of everyday life, in the opinions of others, in trivial concerns and distractions. We treat death as something that happens to other people, something to worry about later, something distant and abstract. We know intellectually that we'll die someday, but we don't really confront what this means. We flee from the anxiety that comes with truly facing our finitude. This flight from death, for Heidegger, is understandable but ultimately a kind of self-deception. It's living in bad faith, evading the fundamental truth of our existence. We immerse ourselves in the they, the anonymous crowd, following along with what one does, what people think, what society expects. We let others define who we are and what matters. We drift through life without really taking ownership of our existence. But Heidegger thought we can choose to live differently. We can acknowledge our mortality, not just intellectually, but existentially, facing it squarely as a constant feature of our being. We can live in explicit awareness that our time is limited, that this existence we have is temporary, that we are finite beings in a world that will continue without us. This confrontation with death, according to Heidegger, is the key to authentic existence. When we truly grasp that we will die, when we face our finitude honestly and without evasion, something shifts. The trivial concerns that usually dominate our attention fall away. What truly matters becomes clearer. We're freed from the tyranny of the they, the anonymous others whose expectations and judgments usually govern our choices. We become capable of taking ownership of our lives, making our own authentic choices based on what matters to us rather than on what society tells us should matter. This doesn't mean living in constant, morbid contemplation of death. It doesn't mean being depressed or paralyzed by awareness of mortality. Rather, it means allowing that awareness to inform our choices, to sharpen our sense of priority, to motivate us to live deliberately rather than drifting through life on autopilot. It means recognizing that every moment is precious because there are only so many of them, and that how we spend our limited time actually matters. Heidegger called this authentic mode of existence being toward death. Living with full awareness of our finitude, making choices in the light of that awareness. It's a kind of liberation, paradoxically achieved through accepting the very limitation we usually try to deny. By facing death, we become free to really live. The Danish philosopher Søren Kierkegaard, who lived a century before Heidegger and influenced his thought, made similar points but from a more explicitly religious perspective. For Kierkegaard, thinking seriously about death is not morbid but potentially liberating and even transformative. It forces us to take our existence seriously, to recognize that we are responsible for who we become, that we must choose ourselves rather than simply accepting the ready-made identities offered by society or inherited from our upbringing. Kierkegaard emphasized that we must internalize death, must think about it as my own death, not just as an abstract fact about human mortality. When I truly grasp that this particular person will die, that this particular existence will end, it should transform my relationship to life. I can't treat my life as an infinite resource to be squandered. I can't put off indefinitely the project of becoming who I'm meant to be. I can't hide in the crowd forever, letting others make my choices for me. This awareness of mortality should drive us to live more earnestly, more passionately, more fully engaged with the moments we have. It should inspire us to confront the fundamental questions of existence. What do I really value? What gives my life meaning? Who do I want to become? How should I live? These are not questions we can delegate to others or find answers to in conventional wisdom. We must each work them out for ourselves, and the awareness of our mortality gives urgency to this project. Kierkegaard was careful to distinguish this from mere morbidity or depression. He wasn't advocating painting your nails black and hanging out in graveyards listening to sad music. Rather, thinking about death should be a motivating force, something that energizes us to take action in life, to engage more fully rather than retreating into despair. Death should be thought into every moment of life, not as a dark cloud hanging over us, but as a clarifying lens that helps us see what really matters. The 20th century existentialist Jean-Paul Sartre had yet another take. Sartre agreed with Heidegger that death is fundamental to the human condition, but he disagreed about its significance for meaning. For Sartre, death is not a possibility that authenticates existence, but rather an absurd fact that removes meaning, or at least complicates it. Human beings, in Sartre's philosophy, are fundamentally free. We create meaning through our choices and projects. We are what we make of ourselves through our actions. But death is the ultimate limitation of this freedom. It cuts us off arbitrarily at whatever point it happens to occur. It transforms our life from an open-ended process of becoming into a finished totality, complete and unchangeable. But it's complete in an arbitrary way, finished at a particular moment that has no inherent significance or rightness. If you die at 30, your life is one thing. If you die at 80, it's quite another. But which it is depends largely on chance, on when you happen to die. This arbitrariness means that death doesn't give meaning to life, as Heidegger suggested, but rather introduces an element of meaninglessness or absurdity. Your life's meaning is hostage to when you happen to die, a factor largely beyond your control. Sartre's contemporary Albert Camus developed the philosophy of the absurd around the confrontation between human need for meaning and the universe's apparent indifference to that need. We want our lives to mean something. We want there to be a point, a purpose, a reason for it all. But the universe doesn't seem to provide any inherent meaning. It just is, neither caring about us nor assigning us a role to play. Death is part of what makes existence absurd. In Camus's sense, we create projects, build relationships, make plans, as if we have all the time in the world, and then death comes and erases it all. The universe doesn't care whether we accomplished our goals or not. From a cosmic perspective, it makes no difference. The absurdity lies in this disconnect between our need for meaning and permanence and the universe's indifference and our own mortality. For Camus, the proper response to this absurdity is neither despair nor false hope. It's not denying the absurdity or pretending that there's some cosmic meaning we haven't found yet. And it's not giving up in despair, concluding that since nothing matters ultimately, we might as well not bother. Instead, Camus advocated what he called revolt, a defiant embrace of life despite its meaninglessness, a determination to live fully and authentically even though death will eventually erase everything we accomplish. This might sound grim, but Camus actually thought it was liberating. If there's no cosmic meaning handed down from above, then we're free to create our own meaning. We're not obligated to fit into some predefined plan. We can make our lives mean whatever we choose them to mean. Yes, we're mortal, and yes, death will come, but in the meantime, we have this existence, these experiences, these opportunities to engage with the world and with each other. That's enough. It has to be enough, because it's all there is. These existentialist perspectives, different as they are in their details, share a common theme. Death is not just a biological event at the end of life, but something that shapes the very structure and meaning of human existence. We are the beings who know we will die, and this knowledge is central to what we are. How we relate to our mortality profoundly affects how we live our lives, whether we live in denial or acceptance, whether we're paralyzed by fear or liberated by awareness, whether we find our finitude crushing or clarifying. Modern psychology has explored some of these ideas through terror management theory, developed in the 1980s by social psychologists Sheldon Solomon, Jeff Greenberg, and Tom Pyszczynski. This theory suggests that much of human culture and behavior can be understood as a response to the existential terror produced by awareness of mortality. The core insight is this: we are animals, like any other in many ways, driven by biological needs and instincts. But, unlike other animals, we're aware of our own mortality. We know we're going to die. This creates a profound psychological problem. We're hardwired to survive, to avoid death, but we know that ultimately our efforts will fail. This creates what the theorists call existential terror, a deep anxiety about our mortality that would be overwhelming if we felt it constantly. To manage this terror, according to the theory, we create cultural worldviews that provide meaning, value, and often some kind of immortality, either literal or symbolic. Religions offer literal immortality, promising that consciousness continues after death, that we'll go to heaven or be reincarnated, or merge with some ultimate reality. But even secular belief systems offer forms of symbolic immortality. Through our children, who carry on our genes and our teachings, through our accomplishments, which outlive us, through our contributions to something larger than ourselves, whether that's a nation, a cause, a body of knowledge, or the progress of humanity. These cultural worldviews also provide standards of value, definitions of what makes a life meaningful and worthwhile. By living up to these standards, by being a good Christian, or a successful business person, or a devoted parent, or a contributing member of society, we earn self-esteem, a sense that our life has worth and meaning. This self-esteem, in turn, buffers us against death anxiety. If my life is meaningful, if I'm part of something larger that will continue, then my individual death becomes less terrifying. According to terror management theory, when our mortality is made salient, when we're forced to think about death, we respond in predictable ways. We cling more tightly to our cultural worldviews, becoming more patriotic, more religious, more convinced that our way of seeing things is right. We judge more harshly people who threaten our worldview or live according to different values. We try to boost our self-esteem by demonstrating our worth according to our culture's standards. And we may show increased interest in having children or leaving a legacy, in securing our symbolic immortality. Hundreds of psychology experiments over the past few decades have confirmed many of these predictions. Reminders of death do indeed affect how people think, feel, and behave in the ways the theory predicts. This suggests that awareness of mortality really does shape human psychology and culture in deep ways, often outside our conscious awareness. But terror management theory has its critics. Some argue that it overstates the role of death anxiety in motivating human behavior. Yes, we know we'll die, but most of the time we're not thinking about it, and we seem capable of living rich, meaningful lives without constantly using cultural beliefs to shield ourselves from existential terror. The theory might explain some aspects of human psychology and culture, but it probably doesn't explain everything, and indeed attitudes toward death vary significantly across cultures. In some societies, death is highly feared and denied, hidden away in hospitals, not discussed openly, treated as a taboo. In others, death is more openly acknowledged, integrated into the fabric of life through rituals and practices that maintain connection with the dead and honor the cycle of life and death. Buddhist philosophy, for instance, teaches that attachment to the self and fear of losing that self are sources of suffering. The goal is to recognize the impermanent nature of all things, including oneself, and to let go of clinging. From this perspective, death is not a terrifying annihilation, but a natural part of the universal process of change. Everything arises and passes away. Form becomes empty. Empty becomes form. To fear death is to resist the fundamental nature of reality, and this resistance causes suffering. Many indigenous cultures have rich traditions around death that integrate it into communal life, rather than treating it as an enemy to be fought or a tragedy to be hidden. Ancestor veneration, rituals that maintain connection with those who have died, celebrations that honor mortality rather than fleeing from it. These practices suggest different ways of relating to finitude, ways that don't center fear and denial, but acceptance and continuity. So, where does all of this leave us? We've traveled a long way tonight, from telomeres and cellular senescence to Epicurus and Heidegger, from mitochondria and oxidative stress to the existentialists and terror management theory. We've looked at death as a biological process and as an existential reality. What can we say now, having explored all these perspectives? First, death is deeply embedded in the biology of complex life. The mechanisms of aging, telomere shortening, cellular senescence, mitochondrial dysfunction, protein aggregation, DNA damage. All of these are not accidents but consequences of how life works. The same processes that allow for growth, development, adaptation, and complexity also create vulnerability to time. We could not have the brains we have, the bodies we have, the richness of experience that makes us human, without also having mortality built in. Second, from an evolutionary perspective, death is not a bug, but a feature. It makes room for new generations, for genetic variation, for adaptation to changing environments. A world of immortal organisms would be static, crowded, unable to evolve in response to new challenges. Death serves life, even as it ends individual lives. The evolutionary process depends on the turnover of generations. Third, we may be able to slow aging and extend health span, the period of life spent in good health. Scientific research is making genuine progress in understanding the mechanisms of aging and identifying interventions that work in laboratory organisms. Some of these interventions are beginning to be tested in humans. It seems quite likely in the coming decades, we'll develop therapies that allow people to remain healthy and vigorous longer, compressing the period of decline at the end of life. But we're almost certainly not on the verge of eliminating death or achieving radical life extension to hundreds of years. The complexity of aging, combined with the fundamental trade-offs built into our biology, means that death will remain part of the human condition for the foreseeable future. We can work toward healthier aging, but we can't escape aging entirely. Fourth, death is not just a biological fact, but a profound existential reality that shapes how we understand ourselves and our lives. Different philosophical and cultural traditions offer different ways of thinking about mortality, from Epicurus's argument that death is nothing to fear, to Heidegger's claim that confronting death is essential to authentic existence, to various religious and cultural frameworks that integrate death into broader worldviews about meaning and purpose. Which of these perspectives is right? Perhaps that's not quite the right question. These are different lenses through which to view an inescapable aspect of human existence. Each has something to offer. Epicurus reminds us that the state of being dead is not itself something we'll experience or suffer through. The existentialists remind us that awareness of our finitude can sharpen our sense of what matters and motivate us to live more deliberately. Various cultural and religious traditions offer frameworks for processing grief, finding meaning, maintaining continuity across generations, and situating individual mortality within larger stories about existence. What seems clear is that how we think about death profoundly affects how we live. If we live in complete denial of our mortality, treating death as something that happens to other people, we may drift through life without urgency, without fully appreciating the preciousness and fragility of the time we have. But if we're paralyzed by fear of death, unable to think about anything else, we may be unable to enjoy life or take the necessary risks for growth and accomplishment. Perhaps the healthiest approach lies somewhere in between, acknowledging our mortality without being consumed by it, letting awareness of our finitude inform our choices without letting fear of death dominate our lives. There's a Japanese aesthetic concept called mono no aware, often translated as a bittersweet awareness of the impermanence of things. Cherry blossoms are beautiful in part because they're fleeting. The brief spring bloom is precious precisely because it doesn't last. This acceptance of transience as fundamental to beauty and meaning might be a useful way to think about our own mortality. There's also consolation in recognizing that we're part of something larger than ourselves. We are temporary, yes, but the species continues. The community persists. The conversation goes on. We add our small verse to the ongoing human song and then fall silent. But the song continues without us, enriched perhaps by our contribution but not dependent on our continued presence. There's both loss and comfort in that continuity. And perhaps there's value in simply accepting mystery. We don't know for certain what, if anything, happens after death. We don't know if consciousness continues in some form or ceases entirely. We don't know the ultimate meaning, if any, of our finite existence in what may be an indifferent universe. Various traditions and philosophies offer answers, but honest uncertainty is also a valid response. Living with that uncertainty, rather than grasping desperately for certainty or fleeing from the questions, may be part of what it means to be fully human. The question, why do we die, turns out to have many answers, depending on whether we're asking about biological mechanisms, evolutionary logic, or philosophical meaning. Biologically, we die because complex organisms accumulate damage over time, because cellular mechanisms break down, because our bodies are built with trade-offs that favor early survival and reproduction over indefinite longevity. Evolutionarily, we die because finite lifespan with reproduction is a more successful strategy for genetic propagation than immortality would be. Philosophically, the meaning of death is something each person and each culture must work out, finding ways to live meaningfully in the face of mortality. But perhaps the deepest answer is simply this: we die because we live. Life and death are not opposites, but partners in a dance that defines what it means to be a living organism in a universe governed by time. Everything that begins must end. Every wave that rises must fall. Every breath taken is followed by a breath released. Life exists not despite death, but because of it, shaped by it, given urgency and meaning by it, defined in contrast to it. Tonight, as you drift toward sleep, consider that you are a temporary pattern of organization in a universe that tends toward disorder. You are a candle flame burning brightly for a brief moment against the darkness. You are a river flowing for a time before emptying into the vast ocean. And there is something beautiful, something almost sacred about that temporality. You are here now, alive and conscious, aware of your existence and your eventual non-existence. That's both remarkable and profoundly ordinary, both precious and commonplace. The atoms that make up your body have been recycled countless times through different forms. They were once part of ancient seas, then part of simple organisms, then part of plants and animals, then part of other humans, and now part of you. After you die, they'll become part of something else, continuing the great cycle of matter and energy that is the universe's way of creating and recreating itself. You are not separate from this process. You are the process itself, briefly self-aware, briefly looking out at the world through these particular eyes before returning to the whole. In the meantime, you have this moment, and this one, and this one. Each moment is all there is, really. The past exists only as memory. The future exists only as imagination. All we ever truly have is now, this present instant, and then the next present instant, and the next, until, eventually, the instants stop coming. But while they're here, they are yours to experience, to savor, to live as fully as you can. So rest now. Let these thoughts settle into your mind without holding them too tightly. The universe has been here for billions of years, and will be here for billions more. You're part of that vast unfolding, a brief note in an endless symphony, a single wave in an infinite ocean. And that's enough. That's more than enough. That's everything. Before I leave you to your rest, if this journey through the biology and philosophy of mortality has given you something to think about, or if it's helped you ease into sleep, I hope you'll like this video and subscribe to the channel. These conversations reach others who are looking for thoughtful, calming explorations of life's biggest questions when people like you share and support the channel. Thank you for spending this time with me tonight. Sleep well, dream gently, and know that while we're here, brief as it may be, we are part of something vast and beautiful and strange. Tomorrow will come with its own questions, its own moments to be lived. But for now, rest. Let it all go. You've thought enough about death for one night. Now it's time to simply be, suspended between waking and sleeping, between questions and silence, between existence and the gentle drift toward dreams. Good night.